Okay, so we're starting or continuing our session, and our next speaker is Professor Ute Deichmann, for the second time on the stage. And so for those of you who doesn't know, Ute started off as a biologist in the University of Cologne, and then she moved to the history of life sciences during her PhD with Ben-Gurion in the University of Cologne. Then she became a professor beginning in London, and then moved to Tel Aviv University, then to Jerusalem, then Philadelphia, and then finally here to Ben-Gurion University at 2007, when she opened the Jacques Loeb Center for History and Philosophy of Science. And she does a lot of very interesting work on the intersection, I think, between history and philosophy, specifically of life sciences, and that is also the purpose of the center. And today I'll let her introduce today's talk, and let's welcome Ute to the stage.
Thank you. Thank you, Jonathan, for the nice introduction. My lecture is on data theory and scientific belief in early molecular biology, Paulings and Crick's conflicting theories of biological specificity, protein synthesis, and the solution to the secret of life. So, big data, but in a historical perspective. I have this one. First, some introductory words about the relationship between data and theory, where I use the term theory in a very general way as any universal statement about nature. Popper, forgive me. In the era of big data, many controversies focus on what should come first, data or hypothesis. And we heard about something of that in Jeremy's lecture as well. So this dichotomy raises several questions. First, even if there is data-driven research, we can maybe discuss it at some other point, without prior hypothesis, is there also hypothesis-driven research without prior data? And to what extent are theories determined by data? And if they are not, what are they based on? These questions have a long history, and the answers depend on the philosophical preferences and lie between the two poles of anti-empiricism and empiricism. On the one hand, there is the notion of the underdetermination of theory by data, which again has a long tradition, also goes back to Goethe even, and is best known as the Duhem thesis or Duhem-Quine thesis. The French physicist and philosopher Pierre Duhem suggested in 1900 that in physics the evidence available to us is insufficient to determine what scientific beliefs we should hold, and this was part of his holistic theory, namely that a physicist can never subject an isolated hypothesis to experimental test, but only a whole group of hypotheses. 50 years later, philosopher Willard Quine expanded this idea and included all activities of intellectual activities, including philosophy, saying that all theories are so underdetermined by the data that a scientist can, if he wishes, hold on to any theory he likes. The other end of the spectrum is characterized by a new ultra-empiricism or data positivism, which we have already heard about, that big data scientists claim of restoring the objectivity of science by relying solely on an abundance of data. Like here in the survey study on science and big data, where it's written, in the new era of empiricism, the volume of data, accompanied by techniques that can reveal the truth, enables data to speak for themselves, free from theory. I think this would not be data first, but data only. My goals here are to examine the relationship between data and theory in Pauling's structural theory and Crick's informational theory of biological specificity, and thereby also to highlight a major controversy at the beginning of what became called informational biology. I want also to show that both Crick and Pauling based their views on data, but only on a very few data, and almost the same ones. And third, I want to claim that despite this apparent underdetermination, the theory choice was possible at the time when data was complemented by logic, causal analysis, and broad knowledge. And that in principle, data are important, but taken alone do not determine theories. So I start with Pauling's structural theory of biological specificity and the data on which he bases it, then go to Crick's informational theory and the data on which he based it, then dwell a little bit on Crick's philosophy. He did also not write it to call it philosophy, but I call it philosophy. And then return to the question of underdetermination in the case of Crick and Pauling. So to start with Linus Pauling, I think everybody knows him here. He was one of the greatest chemists, let's say at least American chemists of the first half of the 20th century. He rebuilt chemistry on the new foundation of quantum physics, in particular by through his significant contributions to our understanding of the nature of the chemical bond. And since the 1920s, he became a pioneer in the physical chemistry of biological macromolecules, in particular proteins. And later he discovered two stable conformations of these structures, namely the alpha helix and the beta sheet. He also tried DNA, but he failed utterly, and that he even admitted. He was not great in admitting failures. And I'm right? Yeah, in '54 he received the Nobel Prize in Chemistry, again, this exam, for his research into the nature of the chemical bond and its application to the elucidation of complex substances. Pauling was an early advocate of the idea that biological specificity had a molecular basis, namely the three-dimensional shape of proteins. Biological specificity is the specificity of individuals, of species, and so on, which was considered a fundamental characteristic of life. So this three-dimensional structure, that was the second part of his belief, is created by molding the protein to the template of other proteins, antigens, genes, and that the amino acid composition or sequences are not relevant. As he formulated it in '37, the secret of life itself is how a protein molecule is able to form out of an amorphous substrate new protein molecules that are made after its own image. And he extended this view generally to specificity, saying that templates and the complementarity of molecular structures are responsible for all biological specificity. Pauling's three-dimensional template theory became the dominant view on protein synthesis and biological specificity, and remained so until the late 1950s, and I should say way into the 1960s. So which were the data on which his theory, the two parts of theory, were based? First, the experimental evidence which he himself and Mirsky gave of a two-step denaturation of proteins that led to a loss of their specific proteins. And they concluded that proteins consist of long peptide chains, which was anything but obvious at that time. And these chains are folded to form a particular shape, and when they are denatured, this shape gets lost. Second set of data comes from the fact that there was a multitude of specific antibodies, even against artificial substances, and this was used by others, not only Pauling, to call into question Paul Ehrlich's selective theory of preformed random antibody formation, and this fact also made template theories of antibody generation popular. And here, antigens directly modify the structure of a normal serum protein to render it specific. Pauling himself suggested such a theory that antibodies instruct. So this is supposed to be an antigen. It's supposed to be an antigen. Could be a
microphone, please.
So an antigen, for example, a virus or a surface of a bacterium, and this surface of the antigen instructs the ends to ends of an antibody, which is just a globulin chain, to build finally an antibody specific against this antigen. And what he wrote and what he believed was that all antibody molecules contain the same polypeptide chains as normal globulin and differ from normal globulin only in the configuration, you would say today conformation, and this conformation is instructed to them by the outside agents. A third set of data came from the anomaly of sickle cell anemia, which was discovered in 1910, and a Mendelian inheritance was early on assumed, but it was confirmed in 1949. And I think everybody knows here, this is a normal biconcave disc of a red blood cell, and in the disease you have just the blood cells forming a sickle. And Pauling, who was dealing with blood during the Second World War in war-related research, he had a hunch that this anomaly could be related to hemoglobin. And he started to investigate physicochemically the molecule hemoglobin in both cases, and he found that normal and abnormal hemoglobin differ in electrical charge. And from there he came to a bold conclusion, Popper would be happy, that sickle cell anemia is related to the structure of hemoglobin, and that sickle cell anemia is a molecular disease, the first such disease, caused by alterations in the shape of the hemoglobin molecule. Looking for causes for this changed shape, he also tried to check the composition. Sequence he could not, there was not yet Sanger, the composition, but he did not find any difference in amino acid composition between normal hemoglobin and sickle cell hemoglobin. And so he concluded that both of them are composed of the same polypeptide chains, folding, however, in different ways. How does the gene come in here? And he said genes serve as templates on which are molded the enzymes which are responsible for the chemical characters of the organisms. First, fourth set of and last set of data came from chemical analysis of protein compositions, where hypotheses on the periodicity and regularity of the amino acid composition were proposed by prominent chemists like T. Svedberg. And these hypotheses that proteins were built regularly were also favored by Pauling. And if this had been the case, it would exclude the amino acid sequence from being the basis of protein specificity, or, in the later terminology, from carrying biological information. And this irregularity or aperiodicity was, I think, first really promoted by Schrödinger's book, What is Life, in 1944, where he likened the gene to an aperiodic crystal which contained the code for life processes. Aperiodic crystal, because periodic crystals cannot contain a code or information. Pauling propagated his three-dimensional template theory of protein synthesis at least until 1956, but in fact also longer. And he either did not relate to contradictory experimental data, or he integrated them in his theory as what Popper would call auxiliary hypothesis. Don't want to go into this, but we can discuss it later. So, second, Crick's informational theory of biological specificity and protein synthesis. Crick was a British, or English even, physicist. He began his PhD in London in physics in 1937, but with the outbreak of war, he went to war-related research on magnetic mines to destroy ships and especially submarines. The Germans were not happy about Crick at the time. And after the war, he worked for the naval intelligence. But somehow he was dissatisfied with his work for the military, and he also developed a passion for questions of biology, especially for the molecules of life, in a way like Pauling. And he read Schrödinger, 1944, and he also read Pauling, 1939, The Nature of the Atomic Bond, and he learned organic chemistry and biology by himself. In 1949, he joined Peruzzi's project in Cambridge on the X-ray diffraction of proteins and learned the mathematics needed to transform X-ray patterns into molecular structures. I found here an interesting quotation which he wrote later, that concerning mathematics. He said, "I soon found I could see the answer to many of these mathematical problems by a combination of imagery and logic without first having to slog through the mathematics." His thesis with Perutz was completed in 1954. It was on the X-ray diffraction of proteins. But we know that a year before he published together with Watson his most famous finding, namely the elucidation of the DNA double helix structure. And Crick and Watson published two papers. This one is from April 1953, Molecular Structure of Nucleic Acids. And this is a month later, a little bit more deep. This is just one page in Nature, and this is 4 pages in Nature, Genetical Implications of the Structure of Desoxyribonucleic Acid. Just a difference to Pauling's paper, which was published shortly before, and which was wrong. It was only about the structure of nucleic acids and not on the genetical implications or any biological implications as such. Crick, when he knew, or he believed at least, that their model was right, this is all well known, I repeat it nevertheless, he ran to the pub, they went to the Eagle in Cambridge, shouting, "We have discovered the secret of life." And what was the secret of life? It was the molecular explanation for gene replication and biological specificity, which was soon after called information. I want to quote two sentences from their second publication, Watson and Crick's. And the first is, the sequence of bases on one chain is irregular, and this certainly reflects Schrödinger's aperiodic crystal. And second, it seems likely that the precise sequence of the bases is the code which carries the information. So it sounds so modern, it sounds so well, but what was code and what was information? Information, after all, I mean, the general theory of how proteins are synthesized was Pauling's, that genes provide physical templates for protein structures and not instructions for amino acid sequences. But 4 years later, Crick came up with a completely different concept of information, namely that genes provide sequences which contain the information for protein synthesis. On what data was this new theory based? So, I'm going through the data very quickly. First, it was Pauling's and other works on protein structure, which Crick knew very, very well. Then the experiments about function and structure of DNA, on which he and Watson based their model of 1953. And then there was also the information discourse of the late 1940s. Crick was at the time at the military. He got to know of it only later, and Pauling could have certainly participated in it, because his colleagues Beadle and Delbruck went to meetings of this informational group, but he never ever mentioned it. So during the Second World War, information became a scientific concept through the US Military National Research and Development Committee, members of which were Claude Shannon, who wrote the Mathematical Theory of Information in 1948, and Norbert Wiener, who wrote Cybernetics in 1948. There were meetings 49 in which John von Neumann suggested the gene as information tape that could program the organism, and Shannon calculated that the genetic constitution contains 10,000 bits of information, of a human of information. But all these nice definitions and suggestions were very vague, and biologists could really not use them. So what can you do with the bits of information, or program the organism? So what does that mean? So code and information entered the vocabulary, but they became only vague metaphors and not exact theoretical frameworks, as my colleague Lily Kay has pointed out. And another set of data, this is very important now, on which Crick's theory of sequence was based, were the experimental results questioning the periodicity hypothesis of protein structure. And here in particular Sanger, who suggested that he not only sequenced insulin the first time, as we heard before, but he also suggested early on that the sequences of 4 species are slightly different. And unlike Pauling, who knew about this but did not pay special attention to it, Crick attributed high significance to Sanger's results. It can be argued, he wrote, for example, that vast amounts of evolutionary information may be hidden away within them. However, he also knew there is nothing in the evidence presented so far to prove that these differences between species are under the control of Mendelian genes. So, nice, sequences are important. So the last, fifth and last set of data on which Crick's theory was based was also related to sickle cell anemia. Crick knew of Pauling's research, and he knew that it was a disease where a Mendelian gene altered a protein. It was one of the few cases where that was known. He also knew, or assumed, that Pauling's method was too crude to detect single changes in amino acid composition. And here, and this I found the most remarkable in the story of Crick, he asked Vernon Ingram to check whether the two kinds of hemoglobin also differed in their amino acid sequence. Vernon Ingram was a very fine protein chemist. He was a refugee from Nazi Germany, came as Martin Immerwahr, and I thought, George, that he himself developed the fingerprinting techniques, but you said he took it from Sanger.
I would think so, yes.
You would think so, yeah. So I didn't know that, so I'm grateful for this information. Anyhow, he used this new way of sequencing, and he confirmed after 1.5 years or so that the 2 hemoglobin molecules indeed differed in one amino acid. And so if no, I'm not saying that. And this was certainly the first demonstration of a change in amino acid sequence caused by a Mendelian gene. And no, I wanted to say still. I think Sidney Brenner is entirely wrong to denigrate the importance of Sanger's findings for the sequence hypothesis. I mean, it was really very, very important.
That's what I believe.
Yeah, no, I think this is simply true. Yeah, here we see it. It is so interesting molecularly that the exchange of one amino acid from glutamic acid to valine, which is a hydrophobic acid, leads to the change of properties, and the molecules, hemoglobin molecules, aggregate and clump together, and this changes the properties of the blood cells. This is a normal hemoglobin being alone. So, and this data, this one change in amino acid, together with the other information he had, led Crick to suggest a new meaning, or a meaning at all, for the term of biological information. And it also led him to suggest his general principles of protein synthesis, which was in 1957. And he wrote, "By information, I mean the specification of the amino acid sequence of the protein." Very simple, but very concrete. And he said, it is of course possible that there is a special mechanism to folding up the chain, but the more likely hypothesis is that the folding is simply a function of the order of the amino acid. And the sequence principle says exactly that the specificity of a piece of nucleic acid is expressed solely by the sequence of its bases, and this sequence is a simple code for the amino acid sequence of a particular protein. Here, information has become concrete, it has become testable, and it was an extremely fruitful suggestion. And here, with this sequence hypothesis, genetic information based on sequences received a central place. In biology, And this caused Horace Judson to say Crick permanently altered the logic of biology, which I quoted before. So, what was Crick's philosophy? He did certainly not base his theories solely on evidence, but he wrote, the direct evidence for the sequence principle and the central dogma is negligible. But still, these hypotheses are very helpful to find explanations and to test them, he wrote on. And he continued to write, I shall also argue that the main function of the genetic material is to control, not necessarily directly, the synthesis of proteins. There is a little direct evidence to support this, but to my mind, the psychological drive behind this hypothesis is at the moment independent of such evidence. I was actually struck to read this from Crick, a hard scientist, maybe one of the hardest scientists in molecular biology at the time, that he talks about the psychological drive being more important than direct evidence. And so I tried to understand what did he mean by this, and he cannot object now, but I suggested. So first, the little direct evidence was the difference of one amino acid in a genetic disease. And the psychological drive, I think, was knowledge of the fundamental importance of proteins in the cell and the species specificity of their sequence, the inadequacy of template theories of protein synthesis, and the uniqueness of sequence in DNA structure, and DNA, the knowledge that DNA was the genetic material. Then, second, this drive was logic and vision. He knew about the dilemma of protein synthesis, that it must be radically different from that of other molecules, because if proteins, so they are synthesized by proteins, enzymes which are also proteins. So these enzymes are synthesized by other proteins. They are also other enzymes which are also proteins. So there is an infinite regress. And so something has to be there. And if this is such a crucial, enzymes are so crucially important for the cell, it must be the genes which must be first. So it was a logical conclusion that it doesn't work like with other molecules such as sugars. So it must be different than that of other molecules, namely highly specific and in all probability controlled by the genetic material of the organisms, which he knew now or believed now that it was DNA. So this to his psychological drive, and later on he wrote about what I call his philosophy, that it is thus not sufficient to have a rough acquaintance with the experimental evidence, but rather a deep and critical knowledge of many different types of evidence is required, since one never knows what type of fact is likely to give the game away. So my fourth point, underdetermination, Pauling and Crick, both based on almost the same few direct data, Pauling and Crick proposed two different theories of biological specificity and protein synthesis. These theories consisted of several parts. In Pauling's case, first, the specificity is based on protein shape. Second, it is three-dimensional templates, not sequences, which determine the shapes. Crick, the specificity is based on the sequences, base sequences, and then amino acid sequences, and the amino acid sequences determine the protein shapes. As we easily see, Pauling's first part was simply complementary to Crick's theory. It was not opposed to it from our point of view at all. Only his template theory contradicted fundamentally the relevance or the importance of sequences. But unlike in the Duhem-Quine underdetermination, the parts were testable independently from one another, as I said before, and they were tested independently from one another. And second, Pauling's theory, the theories were not equivalent, so one could make a choice between them, even though they were underdetermined by evidence, if you wish. So they're not equivalent because Pauling's theory lacked a causal connection of protein synthesis to DNA sequences, which was at that time possible and should have been done. Pauling suppressed evidence questioning his template theory, and when we look at Crick's second part, the folding of proteins as a consequence of a sequence, it was not based on any evidence, not even on a little evidence, but it also did not contradict evidence. So this was indeed a wide speculation, which later on was shown partly to be true, because as you said, George, we don't really know until now exactly how protein fold. I mean, sequence is very important, but there were also still other factors. So a few concluding remarks. Where's Jonathan? Have I? Okay, good. First about scientific belief. It's in my title, so I should say something. Pauling's belief in chemical complementarity and templates was a driving force behind his lasting contributions to the structural basis of molecular biology and medicine. It was really very, very important for something that is important until today. But it also, because it was combined with a very uncritical attitude, it also contributed to his failure to appreciate Schrödinger's theoretical concepts of aperiodic crystal and code, Chargaff's and Sanger's demonstrations of the randomness of sequences in DNA and proteins, and of the central biological question of how biological specificity is generated by the genome, or let's say rather by DNA. Second conclusion brings Crick and Pauling together. This is the inseparability of structure and information in biology. I think unlike Aristotle, who considered form, which we can define, I mean, here are philosophers, but I define it right now as organisms' definition or their essence, as an immaterial principle. Information in modern biology cannot be separated from matter. Information does not exist without DNA or proteins, and the other way around, chemical changes of this matter affect also the information. So structure and information belong together. And Crick wrote in his autobiography a long homage on Pauling, and he summarized it by saying, without understanding molecules, we can have only a very sketchy understanding of life itself. So sequences is not all. So now sequence as not only coding for protein synthesis. Crick later wrote, it was in 1970, about his sequence hypothesis. I should have said that not all nucleic acid sequence must code for proteins, because part of the base sequence can be used for other purposes, such as control mechanisms, to determine if that particular gene should be working and at what rate, or for producing RNA for purposes other than coding. This was 1940. And this somehow resonates, I think, with what Eric Davidson said, who 25 years after after this had examined in painful experimental research what the sequences are doing in terms of the regulatory genome, meaning coding for transcription factors and other regulatory proteins. He wrote, "There is a fascinating world of logic and mechanism that originates in the program of the genome. And I'm not talking about encoding proteins. I'm talking about how the shape of the regulatory network modules determines its function. And here the shape is not just the three-dimensional shape of proteins." Yeah, and here I come to the end. Pauling's theory of the importance of protein shapes and Crick's sequence principle had a long-lasting impact on biology. They were supported not only by a little direct evidence, but also by vision, knowledge, and logic. They were confirmed in their core, not in everything, but in their core, experimentally to today. And I again quote Judea Pearl from his book of Why about big data, where he said, "Unlike correlation and most other tools of mainstream statistics, causal analysis requires the user to make a subjective commitment. Data alone cannot make up for the lack of scientific knowledge." Thank you.
Ute, for being precisely on time.
I tried.
34 minutes. I think I limited to only 2 or 3 questions to get back some time from the beginning. Any questions?
Ute, you didn't talk very much about the psychology of the individuals. What allowed, for example, Pauling to have the presumption to make all of these wild speculations on the basis of little data? Other people are much more conservative. Why did he have that chutzpah to propose all these things that were so disconnected from actual empirical science? That's an important component of understanding why things went the way they did.
You are right. I did not talk about that, because to talk about Pauling's psychology is really a wide field. And I agree that he was not only driven by a vision based on some evidence, but he went far beyond. And in this early part, actually, there was even evidence. I mean, this denaturation, for example, and the sickle cell anemia, there was evidence, and it was great how he speculated from this little evidence on something that became really, really important. But later on, he came up with the things which are far less nice and far less useful. I mean, this vitamin C is one of them. And to carry on this template theory when it was long disproven and influencing so many people with it, I think it is unforgivable.
An amusing anecdote is that Pauling and Erwin Chargaff took a boat from Liverpool over to London, over to New York, sometime in around 1950, 51, 52. And Pauling found Chargaff, who discovered that A equals T and G equals C, to be unpleasant and tedious. And so he avoided talking with Chargaff, and had he talked with Chargaff, his entire view of what the DNA was would have been profoundly shifted. Again, psychological issues interfering with the rational discourse of science.
Right, but on the other hand, Pauling was confronted with a lot of contra-evidence, but he simply suppressed that. Yeah. And he might have suppressed also Chargaff's notions, but it's a very nice story.
This isn't a question, it's a comment, because both my wife and I had the privilege of hearing Crick talk at various meetings we attended. So I wondered if I could share some quotes from him. So one of Crick's favourite quotes was, "If you want to study function, study structure." And another quote is, "Don't let a bad experiment spoil a nice theory." So I mean, these comments, I mean, your analysis of Crick is entirely correct, and I was thrilled to hear how detailed and how well you did it. And these quotes simply complement what you say and back it up entirely.
Yes, concerning psychology. I mean, Crick was, he sometimes he writes those things, and he went far beyond evidence also. But he was really able, I mean, Alan, you can correct me, but from what I've read, he was really able to be self-critical and to take back what was proven wrong.
I didn't know him. I mean, I met him several times.
Yeah, okay, but I didn't meet him even once, yeah. There is, Jeremy?
Yes.
But I hate to raise a historical point with a historian, but I'm amused by how often Schrödinger is mentioned as the originator of the idea of an aperiodic crystal, but it seems to me that Hermann Müller had said as much quite a long time beforehand in a more biochemical way of speaking, and that it was Müller's discovery of radiation as a form of mutation that ultimately led to the experiments that Schrödinger does describe in What is Life. And I'm puzzled as to why it is that Schrödinger, who doesn't refer to Müller directly, he refers to Delbruck and Timofey Rosowski's later experiments, but all of those experiments were really driven under the influence of Müller's sort of vision of how heredity must be functioning. And I'm puzzled as to why it is that Müller has somehow been erased from our collective memory and we remember Schrödinger.
I'm not sure I agree.
Okay.
I mean, Müller is cited in other contexts very, very well. He's known very well. But I don't think he came up with anything. He said we should grind why can we not grind the virus as a model of a gene in a test tube? The first time to suggest we can analyze the gene biochemically, but he never did any of that sort. And I don't know that he never ever mentioned anything of aperiodicity or irregularity in connection with genes. So he did the mutation and he showed that there was a change, but he did not relate it also to molecular composition of the gene. Or I don't really know exactly why you... And I'm not sure Schrödinger knew Muller. He knew so little of biology. He relied on Darlington, on his wrong theory of genes are proteins, and he didn't really care much. He relied mainly on Delbrück's model.
Delbrück got his model from Muller. We should talk about it after.
Okay.
Actually, there is an answer. This is the period, this is the 50s. Muller is a communist in Texas, and he is pro-Stalin, so he is a no-no to mention in polite company.
No, I wouldn't say that. Okay.
So the story of it is that Schrödinger got the idea of aperiodic crystal from Delbrück, and Delbrück discussed it with Timotei Rysovsky, and the idea was the first time expressed by Peltsov in Russia in the 20s and 30s. So before Rysovsky and left the Soviet Union to work in Germany. So the ideas of aperiodic crystal existed even in the book form before.
But sorry, but Delbrück did not write about the aperiodic crystal.
He did not write it, but he was exposed to ideas and
talking?
he told about these ideas to Schrödinger during his visit to Ireland.
Okay, but he did not write about it at all. He was the first to suggest that the gene is something like a molecule.
Absolutely, but these ideas were absolutely integral to the life of the Berlin group that focused on the structure of the gene. And a lot of things were so ephemeral, and so the physicists hesitated to write about them. And it's only the gravitas of Schrödinger permitted him to put it in the written form and promote it. So
that's true.
Yeah, so it's
But-
only...
Sorry.
So it's only until the evidence from the phage group appeared that the gene was understood and had the, and one could point to its molecular substance, right? That these ideas became not just something that you discuss over tea.
That's true, but Delbrück hated the idea of genes being biochemical entities. He hated biochemistry. And no, it's simply true, and that's why he's...
approached it genetically, but in
Genetically, and he still hoped to find new laws of physics by studying the phage, but he was not at the beginning of the molecular analysis of the gene. So
this is why these meetings are great.
We have a discussion tonight, I think, so maybe we could go back and look
Okay.
at the books and get an answer by tomorrow about these subjects. And let's thank Ute again.